a couple of stories to share today about Gencove and what we do. And I've got this very mesmerizing video slide here with things that are not cows, but we will only be talking about cows for the rest of the presentation. So I'll be providing a bit of background introduction on who Gencove is and what we do, what our core technology of low-pass sequencing is, and then go through a couple of applications that we've worked on in the cattle industry. So Gencove got started a couple of years ago out of the New York Genome Center. Our CEO, Joe Pickrell, is a statistical geneticist. Our CTO, Tomas Barisa, computer scientist. And the core of what we focus on is building software solutions to support high throughput, affordable sequencing. We'll talk a little bit more about what that sequencing is and looks like in a second, but in short, we focus on molecular technologies that are very scalable and provide genome-wide information for any organism or any animal or plant anyway. And then we built software and engineered cloud-based solutions to analyze that data and return it to a user in a way that's easy to access and is scalable. And we built our solutions on AWS's cloud platform. So we are very lucky to have had a number of partners in the space. Key amongst them in this area is, of course, the folks at Neogen, and we've been very lucky to be venture backed by groups that are focused on agriculture as well. So we do both human genetics and agriculture, and I lead our product development efforts around agriculture. And in the broader animal space, we've had some excellent collaborations with USDA and folks like ST Genetics, the CTLGH at the Roslin Institute, Agrigen, and Chagas in Ireland. The kind of scope of what we work on has covered now over 25 different species, including fun things like coral, but also your standard things like pigs and cows, dogs, and sheep, and corn and soybeans. We have processed with partners over 250,000 samples to date, which is pretty good for a young company, and have worked with our partners to allow sequencing, which is a technology that is not usually associated with this kind of scale of over 4,000 samples in a day, and can build a customized solution for a new partner for a new population or new species in just a few weeks from a bioinformatics perspective. So what is low-pass sequencing, this core technology that we build around? It involves shotgun sequencing, so this technology of next-generation short-read sequencing of an individual of interest to a coverage of less than 1x. So that means that we get a readout of an individual's genome on average less than one time per position in the genome across the whole genome. And that means that we have a very broad coverage of the information of the genome compared to, say, the 50K array, which only looks at 50,000 positions. We're looking in total at hundreds of millions of positions, but there's a little bit of noise associated with it. So we process it in the framework of what's known as a haplotype reference panel, where we characterize the population diversity of our population of interest. We'll talk more about how we do that in cattle in a bit, and then take those short reads and perform a process called imputation so that we get an accurate genotype call at every single position that we have previously observed in a population. So for example, in humans, we have a reference panel with close to 70 million SNPs. When we do low-pass imputation, we'll make a genotype call at all of those. We see the same thing when we work in cattle or dogs. And all that processing takes place on our software platform. So what this can be used for is trait detection and genotyping, applications like genomic selection, identification of breed composition, mapping of QTLs, and all of the kind of core standard use cases of genotyping tools that you're familiar with. When using the Gencove platform, you have to start by developing a reference panel. So we've worked a lot with many of the folks in the community and using public resources, for example, in cattle. including with Warren on developing a cattle reference panel. And then to apply that, you need to generate some low-pass sequencing data, which is where a group like Neogen comes in. And then we impute those genotypes and help perform any additional analyses. So low-pass sequencing is great because it really provides you with genome-wide information and allows you to easily select different markers. It's very fast and easy to set up compared to designing a new array, and it can streamline the generation of data and provide really fast turnaround time. And it's easy to integrate new markers that you might detect in a population or potentially even work with new reference genomes. We'll hear about some of that today in Ben's talk later. And overall, it provides more value for of data and information for the amount of capital and cash that you're investing in generating these data sets. The platform itself provides a really user-friendly turnkey solution. It's based on the cloud. It's enterprise-grade software that provides security and scalability, and it's based on standardized genomics workflows, and it's been built by geneticists to support and focus on low-pass sequencing. So it's really modern software platform for supporting kind of scale of genomic data that we're increasingly going to be seeing in livestock applications. So I want to go in a little more depth on what we do in the cattle industry and with folks like Neogen, but talk for a bit about some of the validation work that we've done in cattle. So currently now our cattle reference panel is delivering 2.2 million SNPs for every sample that we're genotyping with the folks at Neogen, and those have been carefully selected to have 3/4 of a million of functional variants. So these are variants that impact genes. So this goes way beyond the scope of any array that is being used by the industry today in terms of the content of variants that may be impacting the performance of cattle that we're looking at. And it also encompasses sort of the superset of arrays that have been utilized in the industry to date so that it can be backwards compatible with any genotyping program or breeding program that's out there in the cattle industry. And it provides a very broad representation of the important cattle breeds so that we're able to make imputation in Angus or Hereford, Holstein, Jersey, Brahman, Brown Swiss, Limousin, Charolais, etc. And if I've left out your breed of interest, that's only for sake of brevity here. I know there's a lot of diversity in cattle breeding, and with over 1,800 samples in our cattle reference panel, we're really able to cover a lot of them. We worked with many different groups now to validate this technology, both in cattle and in other populations. This comes from a publication we did with Warren and the Mark team a couple of years ago now, showing that when we genotype animals on an array and we genotype them with this low-pass sequencing technology at half-X coverage, we see extremely high concordance between the genotypes derived from the array and the genotypes derived from the low-pass sequencing. And that was true across several different breeds that we looked at. And even though, for example, there's been more high coverage whole genome sequencing in our reference panel of Angus cattle, we still see very similar performance with Simmental or Hereford. So I want to talk about an application that we've worked on recently with the folks at Simplot to develop an EPD for bovine congestive heart failure as a nice example of how this density of data allows you to tackle the kinds of challenges that we have in the beef cattle industry. So just a little bit of brief background on the trait. BCHF, which I'm sure many of you have heard of, is something that Simplot has seen as causing outsized mortality in their large feedlot facility, which they're then able to detect at a necropsy level as being associated with cardiac failure and cardiac distress, and crops up in the summer and can cause death loss of dozens of samples a week in severe situations. And they have utilized the heart scoring system that was developed elsewhere to allow their vet team to go in and perform necropsies on these animals and determine animals that were severely affected by this trait. And with them, we genotyped close to 5,000 samples out of a total phenotyped population of close to 20,000. And Gencove then took about 1 million SNPs that we identified with low-pass sequencing and ran some modeling on that. And what we found is that, and the great thing about low-pass sequencing and our inclusion of millions of markers is that, if there was a single gene that was the dominant cause of this, you would see a very clear signal for that. We'd be extremely likely to be including it here, and we'd have great resolution for it. And we can see some potential genes that may be contributing somewhat to this trait, but I think we can confidently say that this is a polygenic trait and that we've really captured a clear polygenic signal where we see a high heritability, or moderate heritability anyway, for this trait of somewhere between 30 and 40%, depending on our inclusion of contemporary groups in the model. And we also looked at some markers that had been previously analyzed in the academic literature as associated with this trait, and we're able to see how well they replicated. And so with the versatility of this marker set, we're able to easily sort of compare to other genotyping efforts that have been made in the past. We also see, that this tool genotypes very well across a broad diversity of samples in this commercial cattle population that Simplot has been running through their feedlot. This includes beef on dairy animals, it includes Charolais animals, Angus animals, and all kinds of other commercial beef animals from the Pacific Northwest. And we also were able to see that this trait occurs across the span of the ancestry groups observed in this population. For those of you who aren't familiar, this is a PCA plot that shows genetic diversity in different populations. And so here we see that these cases are observed across the entire span of this data set. So we're also, with this data set, able to show genetic correlations of this trait to a variety of important traits that are under selection and under heavy weighting in genomic evaluations today, and indicate that incorporating BCHF into a selection index would have a major impact on selection decisions in the cattle industry today. We see that growth and dry matter intake are strongly correlated with this phenotype. And so what the Gencove team then did was build a genomic prediction model where we performed 5-fold cross-validation, and we were able to get a correlation between the genotypes, the genetic values that we were predicting for these held out animals and their phenotypes of about 35%, and shows that this low pass data is great for generating genomic prediction models. It also allows us to potentially versatilely use any SNP array that an animal has been genotyped on to evaluate in this model, because we have those SNPs incorporated into the model that we built. So we can make comparisons with any external data set, and we now have a tool that can be used for making genomic selection decisions by Simplot or others to improve the population's incidence of this trait. And as you can see, there's a sort of a strong distribution across the population, where some animals are severely affected by, or not affected, but have a high propensity, and their offspring would have a high propensity for this trait, whereas others have a much lower propensity. And we also see a strong distinction between animals that were the most severely affected, the 5 scores on the far right, versus those that are only slightly less affected, the 4s. And of course, the mortality was most strongly observed in the 4s as well, in the 5s as well. So that's a great example of a demonstration that the technology can work for the core use case of the cattle industry, developing breeding tools and genomic selection. We now have this partnership with Neogen to genotype cattle globally, and we have been working to refine beyond low-pass sequencing to offer a more powerful solution that specifically addresses not just development of genomic selection tools, but also the important diagnostic genotyping that the cattle industry does every day, whether it's for parentage or traits like coat color, pulled status, etc. And we launched this last month, and we're really excited about it. What it involves from a technology perspective is a further addition on low pass of a technology known as hybrid capture, where in a single molecular reaction, we can provide both genome-wide coverage of all the markers that are important, as well as a more refined and high accuracy look at specific targets of interest. So we can, instead of getting a single read across the whole genome, we can take a target like the coat color alleles and get much higher coverage at those positions to ensure that we always very accurately genotype those technologies. And the beauty of this is that we're still able to generate just a single molecular reaction, which results in an output that you see in the middle here, where we still get genome-wide data, but then in specific targets of interest, we get a very high level of data. So that's somewhere between 30x and 100x coverage at our target, which gives us the ability to very accurately genotype that sample. And then all of that processing still takes place on the Gencove platform to do both the imputation for the genome-wide information and the variant calling for our specific targets of interest. The beauty of this is that we're now directly observing these alleles. So, with probes from arrays, for example, we're sort of making comparisons between two known positions, but with the sequencing data, we can really directly see, and I'll show you in a second what that looks like, exactly what's going on at that location. And of course, that can have a lot of advantages to continue to monitor in our populations what's going on. It has more accuracy than existing genotyping tools, high coverage, whole genome sequencing. When successful is really the gold standard of genotyping in the modern genomics toolbox. And there's a lot of flexibility for continuing to expand the marker set that we're developing using this, because this technology of hybrid capture, where we target specific regions of the genome, doesn't rely on rebuilding chips the way we're familiar with iterating technologically in the cattle industry, where we have to update our chips when we want to add new traits or disease alleles to a SNP array platform. So we set out with Neogen to design this hybrid capture probe set at over 800 locations that are important to them, including things like parentage and the proprietary diagnostics that Neogen customers rely on. And we validated this in several hundred samples that are known carriers of key alleles with chip genotypes. And we were quite successful in our design process for validating this, meaning we were able to get most of the targets that we set out to capture to generate high coverage data in our initial run. And that means that we have a lot of flexibility to successfully ensure that we get information about these targets with this technology. And if somebody comes along and says, okay, we have a new disease in this breed of cattle, or a new trait associated with a new QTL associated with some trait that we're trying to breed, we'll be able to incorporate that into this target capture very readily from a technical perspective. Some of the examples of samples that we processed in the first few hundred targets were the polled allele, MC1R, A2, and calpain. And yeah, we saw that we were able to get, in most of these cases, very comprehensive concordance between the genotyping that we did on arrays and the data that we were seeing from the high coverage data. So to give you a deeper sense of what this data ends up looking like, these little bars represent the reads that we're generating with sequencing and their position along the genome. And you see that in the center here, we see high coverage data, where we're detecting sort of a position that's the reference position in blue, and then red, the alternative allele. This is sort of an illustrative figure here. And so we have three different individuals here, and the bottom one is alternative. The middle one is heterozygous, and the top one is carrier of the ref allele. So we've truncated this, but each of these bars represents a single read of information. And so at each of these individuals, we'll observe more than a dozen times the two alleles present and be able to confidently make a genotype call at that position. Whereas, what you see around it is that low pass data where we're relying on imputation to make our accurate genotype calls. And this means that the accuracy of this can be much higher than 99%. This also allows us, for example, to directly observe deletions, like the deletion that causes the Celtic polled allele present in Angus cattle. And here we see what that 16 base pair deletion looks like with the target data that our panel generates. So in summary, Gencove's low-pass solution provides really high-quality data that is easy to integrate into the kinds of applications and solutions that the cattle industry is using today, and can be an affordable replacement for arrays. And we're working across the industry to help folks figure out how to best use this data today, and are excited to continue to see where we're able to take sequencing and genomics in cattle. So thank you all for your time and attention today, and